Okay, so this is the first video of cellular biology. So this is introduction to the cell. This is going to be a relatively short video, and it's going to be just a very general basis of what the cell is, the components of the cell, and from here then we'll continue on into a deeper dive into molecular, into metabolism, but everything starts off at the cell. So the first thing we're going to talk about is the cell. Why is the cell the foundation of medicine? And the reason it is because it is what we call the smallest unit of life. So if we were to look at all the living things in the world and you were to look at the smallest component of it, we call that component the cell. What's important to recognize is that the cell has components within it. These components in of itself are not what make up the smallest component of a living organism. Meaning, if I were to look at just the ribosome, a ribosome in itself does not make the smallest component of a living organism. A ribosome within a cell, a cilium within a cell membrane, a rough ER, a nucleus, all of these components together are what make up the cell, and then the cell makes up that smallest unit of life. But what is important to know are these components. So you should have a basic understanding of these components. You should know that ribosomes, which we will talk more in depth in a future video, but ribosomes, eventually when we get into it, you should know that those are the protein builders. You should also be aware that the cell membrane is the protective layer of the cell. We will eventually learn how to be familiar with terms like semi-permeable. Semi-permeable is what allows the cell to be protected because it allows certain things to come through it and some things it doesn't allow. We should also be familiar with mitochondria. That is the powerhouse of the cell. So a lot of your ATP generation will happen from here. Smooth ER. We will talk about it in future videos, but steroid hormones related to smooth ER. Nucleolus, nucleus. What's the difference between both? Nucleolus will have to do with our RNA. Nucleus has to do with your genetic material. So all of these components, and notice how when you're looking at different components of the cell, everything has to do with a different function. So when you're looking at mitochondria, you're talking about ATP. So metabolism, or a major part of metabolism, happens in the mitochondria. When you're looking at the smooth ER, you're talking about steroid hormones. So hormone production, that is happening at the cellular level. And when we're talking about steroid hormones, we are talking about hormones like FSH, LH, testosterone. All of these hormones at a cellular level are being generated. What are these hormones leading to? FSH, LH, that helps you and helps women with their menstrual cycles. It controls the menstrual cycle. Testosterone, that is the male-dominated hormone. So all of these hormones together are, being generated in the smooth ER. So your smooth ER has to do with hormonal production. Nucleolus, rRNA. rRNA has to do with ribosomal production. So notice how different components of the cell have to do with very different functions. All of those functions together allow the human to survive. So that is the point of that. We will get into each of these in a future video. So this is why it's important to study the cell, because everything has a certain function. Now notice how these functions within the cell lead to bigger functions of the overall organism. So every disease in medicine also begins with something wrong at the cellular level. So if we're talking about cancer has to do with abnormal cell division. you can see like cancer has to do with an issue with proto-oncogenes or tumor suppressor genes. So this is happening at the cellular level. It's the same thing with diabetes, abnormal cell signaling. So if we're talking about diabetes, we can talk about GLUT2 and GLUT4 receptors. So what part of the cell do GLUT2 and GLUT4 have to do with? That's going to be your cell membrane. So understanding things at the cellular level will help you understand things and it will help you understand diseases that we hear about in our everyday life. So that is something, and that is a reason why studying things at the cellular level is very important. So now we talk about the cell theory. The cell theory is, if you are studying for the MCAT, it would be good to know it. If you are past the MCAT and in med school, it's probably not something you will directly be tested on, but nevertheless something good to know. So all living things are made of cells. The cell is the basic unit and structure of life, and all cells arise from pre-existing cells. The reason this third one is important is because back in the day, people used to think that cells came from things that were non-living. So the cell theory was one big change in that pre-existing notion. So cells are part of living things because they undergo metabolism, reproduction, homeostasis, growth, and response to stimuli. Now, the reason this is important is because cells can undergo these processes and they don't need another host organism to undergo these processes. So every cell can do that individually. This is in contradiction to viruses. And that is why we consider viruses non-living organisms. Well, non-living, sorry. They can't be organisms if they're living. So that's why we say viruses are non-living. Because they need a host to allow replication of their genetic material to happen. They cannot replicate. They cannot make their own food. They can't do anything without a host. But our own cells independently could do things. So now we're talking about biologically important molecules of the cell. So when you're studying for the MCAT, this is going to be part of the topic that has to do with 4 macromolecules. Now we will look at each of these in depth. But proteins, this has many diverse functions throughout your cell and throughout your body. Carbs is your main source of fuel. It's your quick energy. What is a very good example of a carb? Glucose. That's your quick source, your sugar. Lipids are, yes, you can use them for energy, but they're more for long-term energy because they're stored in the form of fats. Lipids also are found in your plasma membrane. So they help protect your cell. Nucleic acid, aka your genetic material. You see it in the picture here, you see it in the background. But this is what basically the genetic material is, the genes that you get from mom and dad. So let's take a look at proteins. So the same way we say that the cell is the smallest component of a living organism is the same way we say that amino acids are the smallest component of a protein. So amino acids are the building blocks of proteins. Very important for the MCAT to know the structure of an amino acid. You should know that there is an amino group, there's a side chain, there are 20 amino acids, so therefore you will have 20 different side chains, but the basis is the same, meaning this backbone is the same for all your amino acids, and then this is what changes. Your side chain is what changes. So if we were to look at a side chain that had an H, so say your R group was an H, that means your amino acid would be a glycine. Another thing to note about amino acids, this is not, again, this is like an introduction, but just to go into a little bit of detail, this is what we call a chirocarbon for 19 of your amino acids, because 19 of the 20 amino acids have R groups that are not H2N, H, and COOH. And chirocarbon basically means that your carbon is attached to 4 different functional groups. Functional groups or elements. But in the case of glycine, that's attached to 1 hydrogen and the second hydrogen, that is the only amino acid that is achiro. Again, we will talk more in depth about this in a protein lecture. Another thing that's very important to know is levels of protein structure. So the way things work, and I did not touch upon it, but look over here, that when we talk about cells build up and multiple cells come together and form tissues. Tissues, a bunch of tissues, will form an organ. A bunch of organs will form an organ system, like over here. If you can tell, that is your digestive system. That is the liver. That is your large intestine, small intestine. And then over here you have an organism. So cells make tissues, tissues make organ system makes organism. So now when we go to proteins, you have different levels of protein structure: primary, secondary, tertiary, and quaternary. So we will go in a little bit of depth of each of these. So your primary structure of a protein is going to be a linear sequence of amino acids. This is something, if you are studying for the MCAT, memorize this. Memorize it hardcore, because you're very likely to get a multiple choice question that will say, that will use these specific words of like linear sequence of amino acids. So again, if we were to give like an example of this. This is how your primary structure would look. Glycine, leucine, alanine. Just your amino acids being connected to each other by peptide bonds. That's primary. Secondary has to do with folding. But what happens is when you look at your amino acid structure, so let's say this is residue 1, and then you get 2, 3, and 4. Between your first and fourth amino acid, you are going to have a hydrogen bond. Let me first draw it out. You will have a hydrogen bond forming between this and this. This hydrogen bond between these two is going to fold the amino acid, and you are going to get something called alpha helices and beta pleated sheets. So the 1 and 4 residues are going to form hydrogen bonds, and then you'll get this kind of folding. This is different than your tertiary structure, because your tertiary structure, and also another thing important for the MCAT, know how it looks. So your 1 and 4 residue will form the alpha helix. Your beta pleated sheet is different, and your beta pleated sheet, just know that the bond, like the reason it develops a different shape is because like the bonding is different that leads to the folding. So depending on how the bond is between two atoms, two atoms in your linear structure is what's going to lead to the folding. So the type of bond leads to the type of fold. Yeah. Another thing to note is that when we're talking about tertiary structure, this is bonds formed between R groups. So when we're talking about secondary structure, these are bonds formed between the backbone structures. So notice how your carboxylic group and your amino group, they're still within the backbone, if that makes sense. But for your tertiary structure, it's between R groups. That is why when you have, I'll draw out the full amino acid again. And let's say over here you have an SH. So when you have a bond formed between these two thiol groups, that is going to form a disulfide bond. So notice how the bond, and that is part of your tertiary structure. So notice how the bond formation is happening in your R groups, leading to tertiary structure. And that's why you have this fold. So again, it goes back to the concept that the type of bond that's forming leads to the type of bond that is forming between two elements in your amino acids is what leads to the type of fold that you will see. So if you see bonding between your R groups, you're going to see a tertiary fold. But if you see bonding between residues in the amino acid sequence, that in the backbone of the amino acid sequence, that is going to be secondary fold. And then your quaternary structure is going to just be multiple subunits. So you will always see multiple subunits. You will never see something within one protein or within one subunit of a protein. Yes, not within a protein, within one subunit of a protein. That will not be a quaternary structure because your entire protein is going to be made up of subunits. So if we were to just break this down, 1, 2, 3, 4, that's one subunit, that's two subunit, that's three subunit, that's four subunit. What is an example of something that has 4 subunits? Hemoglobin. Very important to know for a biochem class. Now let's talk about carbs. So carbs are your sugar energy source. And carbs are very useful for quick energy because they're easy to break down. What is the main thing we have learned for carb breakdown? Glycolysis, because we intake glucose through food and then glycolysis eventually breaks it down. So when we're looking at monosaccharides are the building blocks of carbohydrates. We have glucose, we have fructose, and we have galactose. Those are 3 examples. Glucose is, again, the main source of energy because we take things like when we eat food comes in the form of glucose. Like when we break down our food, the monosaccharide that comes is in the form of glucose. Fructose is an isomer of glucose. What's important to note about fructose, though, and let me correct that here. Your fructose ring looks like this. Your fructose ring is a 5-carbon ring. It's not a 6-carbon ring. However, your fructose is a 6-carbon sugar. So I'm going to say this, and this is something that is very important for your MCAT. Glucose is a 6-carbon ring, and it's a 6-carbon sugar. Fructose is a 6-carbon sugar, yes, and it's an isomer of glucose. However, it's a 5-carbon ring. And it's the only one of your sugars from the isomers of glucose that is a 5-carbon ring. Everything else will be a 6-carbon ring. Know that for the MCAT. Then you have disaccharides. So disaccharides will basically be forming from an alpha 1,4 bond, an alpha 1,6 bond. So in this case, you will have an alpha 1 and alpha 4 bond. That is not your alpha 1. That's your alpha 1. That we'll talk about in more detail in some other video. And then you have polysaccharides. Polysaccharides are going to be multiple monosaccharides. So disaccharides are 2. Polysaccharides will be multiple. Very important to know starch for biochemistry class. Glycogen is going to be something that we learn in metabolism, but glycogen is going to be the polysaccharide of glucose. Very important to know for MCAT. And glycogen is stored in the liver. Also, it's going to be a step 1 MCAT thing to know. Now, the third macromolecule, lipids. Lipids are something that you will find in different forms. So it's the same thing. Proteins, you find it in so many diverse forms. Lipids, you will also find in a lot of diverse forms. One of those forms is going to be in the form of phospholipids. Phospholipids are found in the cell membrane. So that's what we talked about when we said cell. So the cell membrane is a cell part. Very important to note that you have a hydrophilic head and a hydrophobic tail. And how do they format themselves? Like this. This is what forms your phospholipid bilayer. So, and this is why we say that for something to enter the cell, it needs to be hydrophobic. Otherwise, it would need to go through a protein channel, because this inner layer is all hydrophobic tails. Now, if we were to look at the structure of your hydrophobic tails, that is in the form of fatty acids. Now, this whole structure here is called a triglyceride. This is not a fatty acid. Your fatty acid is just a portion of the triglyceride, which is this part. This part, but sorry. It would basically be a long chain plus a carboxylic acid at the end. Now, try to differentiate between everything. Phospholipid is what you find in the cell membrane. Triglyceride is energy storage. So triglycerides are stored in adipose tissue, in your fat tissue. That's different. Now, triglycerides are made of a glycerol and 3 fatty acids. Fatty acids are a base molecule that make up both triglycerides, which you see here, and it also makes up the hydrophobic tails in phospholipids. So again, fatty acids are a base molecule. Fatty acids make up phospholipids, and fatty acids make up triglycerides. Phospholipids are found in your cell membrane. Triglycerides are found in adipose tissue. They are used, they are broken down after like hours of not eating. So if we take it a step back, when I'm looking at carbs, initially when I want fast energy, I have glucose from my meal. I use that glucose that that I just ate from a meal to give me energy through glycolysis. What happens after is, like, say if I'm fasting for a few hours, then glycogen, my glucose stores will break down. After all my glycogen stores have been used up, then I go into breaking down triglycerides. And these fatty acids will break down, travel through the blood, go into cells, and they will undergo a process called beta oxidation. That is what we will discuss in metabolism, though. This is just a general overview lecture of the cell. Now we're talking about steroids. Steroids are very important for the cell membrane, and an example is cholesterol. So cholesterol has, which we will talk about here, it maintains membrane fluidity. So when you're at a low temperature, your phospholipids like to stick together. So cholesterol comes in here and allows it to sort of build a space between these really stuck phospholipids. So it prevents this tight packing. So that's why at lower temperatures, you increase the fluidity. But notice how at higher temperatures, like cholesterol is over here, but it's preventing all of these from dispersing, from moving completely away from each other. So the fact that it's preventing it from moving away means it decreases the fluidity at a higher temperature, because at high temperatures, there's too much fluidity. So I want to reduce that. So in that sense, it's a maintenance molecule, and it acts in both high temperature and low temperature. So that's something to keep in mind. Terpenes are another class of lipids. So again, just to go back, we talked about phospholipids as an example. We talked about triglycerides as an example. Fatty acids make up phospholipids and triglycerides. They're a component of it. Steroids, we talked about cholesterol. That's an example of a steroid that helps maintain membrane fluidity. There are also other steroids, such as your steroid hormones. Those are lipids, but those are not part of your cell membrane. That's a different topic. Terpenes are also a class of lipids. They are made up of a C5H8 unit. So every terpene, like the base unit, is a C5H8, and that builds up. What I would know for the purpose of the MCAT is monoterpene is 2, sesquiterpene is 3. I would know the number of carbons, and then based off of the number of isoprene units, just multiply by the number. And do try to memorize the names for the basic ones. Now, nucleotides. Nucleotides are the base molecule, the base unit, of your DNA and RNA, so your genetic material. So they are the building blocks of nucleic acids. A nucleotide is composed of 3 components. This is your first component, the nitrogenous base. Purines have 2 rings, pyrimidines have 1 ring. Purines are adenine and guanine. Pyrimidines are cytosine and thymidine. Now, this is your sugar. So DNA has an H, RNA has an OH. That's why you call DNA deoxyribonucleic acid, because it lacks an oxygen that the RNA has. That's why that is called ribo rather than deoxyribonucleic acid. Notice how the 3, the 1, the O, and the 5 are all the same. And then this is 1 phosphate, but when we look at other structures like ATP, you will see triphosphates. So one thing to note about the structure of DNA is when you form DNA from a nucleotide, you form a phosphodiester bond, like a 5 to 3, a 3 to 5 phosphodiester bond. So one thing to note about this, note how this 3, the oxygen here, is going to do, if I were to draw, which I will draw actually, I will redraw this 3OH and pretend everything else in the molecule is the same. And then I'll just draw your sugar. So this 3OH, and then I'll draw another H. And I'm drawing H here because I'm representing a DNA molecule. Now let me transfer this molecule here. Now, what this O is going to do is it's going to attack this phosphate. Because notice how this is relatively electropositive. So this is serving as your electrophilic center. OH, I mean, it would lose its hydrogen, and then your O would become negative. So this would serve as your nucleophile. So your nucleophile is going to attack the electrophilic center, and then you will eventually form a phosphodiester bond. So this is not, so you have your O forming the bond with the P. And you don't have to know the exact format of the whole process. Just know how it's a 3,2,5 phosphodiester bond. Because your 3OH is attacking the phosphate. Notice, however, that your DNA molecule from starts with a 5-phosphate, and then if you end it, you have your 3OH down here. So that's why you say that your molecule, your DNA molecule, is gonna starts from the 5 to 3. But the actual phosphodiester bond runs in the 3 to 5. That's one distinguishing factor between both. That would be helpful to know for the MCAT. So just know phosphodiester bonds, and then know DNA runs in the 5 to 3. But then when you're looking at certain nucleotides, let's zoom in here. Say if I have a molecule that looks like this. If I were to look at these bonds, this and this bond here, so this is an OP and this is an OP. However, this is not called a phosphodiester bond. So notice this is a phosphodiester bond, which we labeled here. This bond between the phosphates, so sometimes you'll look at molecules like ATP. This is known as a phosphoester bond, not a phosphodiester bond. So, key points. There is a phosphodiester bond. Phosphodiester bond will run in the 3 to 5, which I explained based on the nucleophilic attack here. You have a phosphoester bond. That is between phosphates. And then your third is going to be DNA reads in the 5 to 3. And that's because as you keep adding nucleotides, it's going to look like this, which that is a pretty ugly picture. But that is, that's just the basis of how it works. Know these structures. If you are studying it for the MCAT will never make it that confusing. So you don't have to know every element. You just have to have a general idea of how it looks. So you should be aware of the fact that adenine has an NH2. You should be aware of the fact that guanine doesn't have that NH2 that adenine has. But guanine will have the carbonyl. And then cytosine, thymine, and uracil. Just know the differences. That's the main thing that you would want to know for your MCAT. But that is pretty much everything for the intro. Coming up in our next videos, I will post more about prokaryotic and eukaryotic cells. We'll talk more about organelles, go more in depth. I'll give a lecture on the plasma membrane, membrane transport, receptors and cell signaling. We'll get into the G protein coupled receptors, the cytoskeleton, cell junction, cell cycle, which includes mitosis, meiosis. That will form the basis of cancer biology. And then we'll talk about apoptosis, necrosis. So when your cell undergoes something disruptive, or it needs to die, or it has to kill itself because something went wrong. We'll get into that, and then we will get into stem cells. That will conclude our cell biology playlist, and then we will continue into molecular after that. So I hope this video helped, and feel free to leave a comment if you have any questions.